Welcome to Dr. Balaga's podcast for the Curious Doc. Today is June 15, 2026. This video is on a Nature medicine article published today online titled Plasma Proteomic Signatures of Cellular Aging Predict Human Disease. It's an open access article. There's associated content titled Blood Signatures of Cell Type Specific Aging Forecast Disease Risk and Resilience. Plasma proteomics may quantify biological aging at cellular resolution. Study cohort comprised of 60,542 participants across GNPC, UK Biobank, and NSHD cohorts. Greater than 7,000 plasma proteins are measured. Machine learning aging clocks developed for over 40 cell types. Validation across SomaScan and Olink platforms, follow up to 15 years. Statistics, age gap modeling, correlations, hazard ratios, Kaplan-Meier analysis, false discovery rate corrections. What did they find? 20 to 25% exhibited accelerated aging in a single cell type, 1 to 3% in more than 10 cell types. ApoE4 carriers had older astrocytes, younger macrophages, and ApoE2 showed just the opposite pattern. Extreme astrocyte aging tripled Alzheimer's disease risk among ApoE4 zygotes. Youthful astrocytes reduced Alzheimer's disease risk by greater than 60%. Extreme skeletal myocyte aging predicted amyotrophic lateral sclerosis, hazard ratio 12.74 versus youthful aging. Log rank p-value less than 0.00001. The cardiomyocyte aging predicted amyotrophic lateral sclerosis, had hazard ratio 6.59. Astrocyte aging predicted Alzheimer's disease, hazard ratio 12.59. Extreme versus youthful. Log rank p-value less than 0.0001. Smokers with extreme respiratory epithelial aging had 58% higher lung risk than smoking alone. Polycellular aging burden strongly predicted mortality. Limitations of the study: predominantly older individuals of European ancestry. Cell type assignment inferred from transcriptomic mapping. Rare cell populations incompletely represented. Key takeaway: cellular aging is highly heterogeneous. Cell-specific aging signatures forecast disease susceptibility, resilience, and survival. What does the research briefing say? Aging drives greater than 50% of the global disease burden. They describe the study cohort, that is 60,542 individuals from different biobanks, including the UK Biobank. Greater than 7,000 plasma proteins linked to 40 cellular signatures. Machine learning model estimated cell type biological age. What did they find? Accelerated astrocyte aging increased Alzheimer's disease risk. Useful astrocytes were protective. Skeletal myocyte aging conferred a 12-fold higher risk of ALS. Aging T and B cells predicted lymphoma. Combined respiratory epithelial and alveolar type 2 cell aging identified smokers with a 2-fold higher lung cancer incidence. Survival remained around 90% at 15 years with largely normal cellular aging profiles, but fell to 34% among individuals with greater than 20 aged cell types. Muscle lineage aging was the strongest mortality predictor, greater than 4-fold higher risk. The limitations are limited representation of rare cell types. Plasma cell mapping may not perfectly reflect protein origin. Generalizability beyond predominantly European cohorts requires validation. Key takeaway: blood-based cellular aging signatures identify both vulnerability and resilience states. Clinical pearls: Plasma proteomics may enable non-invasive assessment of cellular biological age. Astrocyte aging provides risk stratification beyond ApoE genotype for Alzheimer's disease. Youthful cellular states appear biologically protective, not merely absence of risk. Skeletal muscle aging may precede clinical amyotrophic lateral sclerosis ALS by years. Cellular aging burden behaves as a systems-level biomarker of mortality. Distinct cell types age asynchronously within the same individual. Future preventive strategies may target specific vulnerable cell populations rather than chronological age alone. This is an infographic on this very important Nature article. Hit pause and read at your leisure. Podcast brought to you by masterbedfacts.com. This podcast is for physicians and healthcare providers. If you enjoyed this video, please like, subscribe, and forward to your friends.